So this is part 2 of the lecture for Unit 1. We talked a little bit about bacteriophages, and so now we're going to talk about the bacteria themselves, the hosts for those bacteriophage viruses. Bacteria are single-celled organisms. They are microscopic, so they're too small to see without a microscope. But like all cells, they have a few things. They have a cell or cytoplasmic membrane. The cell membrane is shown here as this gray layer surrounding the cell. And the cytoplasmic membrane is made up of a chemical called a phospholipid. And those phospholipids form what we call a bilayer. We'll talk more about that later in the course. But the cell membrane is to separate the stuff inside of the cell from the stuff outside. And it helps cells in all organisms regulate their internal environment. So inside of that cytoplasmic or cell membrane, there is cytoplasm. And cytoplasm is a liquid that is inside of the cell. And the cytoplasm contains all of the stuff that makes the cell work. It contains nutrients and other chemicals that are necessary for the cell to produce energy. And contains enzymes and structural components that make the cell move and give the cell its shape. Right? So everything that makes the cell work is in this liquid layer inside of the cell. Most importantly, inside of the cell is the DNA, or the chromosome of this cell. So the DNA, or what we call the genome of the bacteria, is the information that contains all of the instructions for making the cell the cell. Also, inside of the cell, you can find a number of other structures, including ribosomes. So ribosomes are large protein and nucleic acid components. They're large structures inside of the cell that are there for making proteins. They are some of the biggest things inside the cytoplasm, and so they can be seen with electron microscopes. And so they're something that we often see when we look at images of bacteria. There are also a number of things on the outside of the cell. Most importantly for bacteria, there is a cell wall that is a rigid structure on the outside of the cell membrane. So the cell membrane is kind of an oily layer around the cell. The cell wall is a stiffer, more rigid structure. It's made up of a chemical called peptidoglycan. Also, many bacteria produce what's called a capsule. The capsule surrounds the outside of the cell, and it's a sticky layer that helps protect the bacteria from the environment and also helps it adhere to surfaces. Bacteria have a number of appendages that stick off from them, including pili, which are small hair-like appendages that bacteria use for a number of things, including sticking to things and also transferring genetic information between neighboring cells. And then shown here, there's a flagellum, which are structures that many bacteria have that they use like whip-like tails that allow them to move through their environment. Right? So this is a pretty simple organism. As we can see here, it's made up of a single cell. It's got a few important components. But biology is often much more complicated than it appears on the surface. So even though this is a single-celled organism, if we looked at the molecular structure of this bacteria, we would see that the cell looks something like this. Right? Those structures in the cell are made up of individual molecules that come together in really complex ways to make the cell do all the things that it does. You can see here this flagella is actually made up of hundreds or thousands of individual proteins that form this structure, including the whip-like tail itself, and then this incredibly complex motor complex that lets the bacteria turn the tail when it needs to and turn it off when it does not. Inside of the cell, we can see some of these ribosome components and also a number of the additional enzymes and nutrients that float inside of the cytoplasm, which is really a rather complex environment with all of these individual complicated chemicals that bacteria need to survive. bacteria in general belong to two different groups that are distinguished by some differences in their cell wall and cell membrane structure. So those two groups we call gram-positive and gram-negative. And so these are two distinct evolutionarily related groups. The differences that are most important are in the cell wall. So gram-positive organisms have a rigid cell wall that's made up of these peptidoglycan layers. In gram-positive cells, there are multiple layers of peptidoglycan that stack on top of each other to form a thick cell wall. So those layers of peptidoglycan are attached together. They're cross-linked by another chemical called teichoic acid, and also by lipoteichoic acids, which anchor the cell wall to the cell membrane. Right? So gram-positive cells have a thick layer of peptidoglycan on the outside that makes up their cell wall. And that peptidoglycan surrounds a single phospholipid bilayer membrane that's shown here. And so gram-positive cells, thick peptidoglycan cell wall, single phospholipid bilayer membrane. Gram-negative cells, on the other hand, so they have a layer of peptidoglycan that makes up the cell wall, but it's a thin layer, so one or two layers of peptidoglycan. That peptidoglycan cell wall is actually between two different membranes. So there is an outer membrane in gram-negative cells. It's made up of phospholipid bilayer with a number of additional chemicals attached in it. And they have an inner membrane, also a phospholipid bilayer. But the space between those two membranes, what we call the periplasmic space, contains the cell wall and also a number of additional chemicals that float in that periplasmic space. Right? So gram-negative cells have a thin layer of peptidoglycan, and that's in between two phospholipid bilayers. So bacteria reproduce by a process that we call binary fission, where one cell splits into two. So when bacteria go through this process, they have to replicate their DNA, they have to produce more of the cellular components, they grow so the cell becomes bigger and then splits into two new daughter cells. Right? So bacteria can undergo this binary fission process rather quickly. So E. coli, which is a common bacteria that we study in labs, E. coli divides when it has plenty of nutrients approximately every 20 minutes. So if we give some cells in a culture plenty of nutrients, they will grow and then divide into two new cells. So after about 20 minutes or one generation, there will be double the number of cells in the culture. Those two cells can then both undergo binary fission after about 20 minutes, and so that after 40 minutes there will be 4 cells. And then those cells can also undergo binary fission, so after about 60 minutes there will be 8 cells. And so we can track the number of bacteria in a culture by tracking how many times they double. So we use this equation to keep track of the number of bacteria after a number of generations or bacterial reproductive cycles. So this equation means that the number of bacteria after n amount of generations is equal to the number of bacteria that we start with, right? N naught or N0 times 2 to the power of the n, or the number of generations. And what this equation really means is that the number of bacteria that we have after a number of generations is just doubled every single generation. Right? So in this example here, we have 3 generations, or 3 divisions. 1, 2, 3. And so we started with 1 cell, right? So n naught equals 1 times 2, that's doubling after 1 generation, times 2, doubling after 2 generations, times 2, right? And that equals 8, right? And we can just rewrite that as 1 times 2 to the 3 generations that we just doubled. So because 1 bacteria after 20 minutes goes to 2 bacteria, and then 2 bacteria after another 20 minutes goes to 4, the number of bacteria can increase rather quickly. Some bacteria take a lot longer than 20 minutes to grow and divide. However, if we look at bacteria growing in a culture, we can track their growth in the number of cells and look at that growth on a graph that will look something like this. So this graph shows the logarithm, or the logarithmic value of the number of living bacteria cells. So this is approximately the number of cells in the culture. So, and on the x-axis here we have time, the amount of time that passes. We put some bacteria into a new culture with plenty of nutrients here. After a bit of time, we won't actually see an increase in the number of bacteria. And that's because this is what we call lag phase. So it often takes bacteria and cells to a little bit of time to adjust to their new environment. And so there's a short period of time, or perhaps a long period of time, depending on the organism, before it actually starts growing. But bacteria, when they're prepared to grow and divide, they double every time they grow. And so their number increases really fast. And this is what we call the log phase, where there's an exponential increase, right? So we have 2 to the power n. That's an exponential increase in the number of living bacteria cells every generation. So you go from 2 to 4 to 8 to 16 to 32 to 64. After a while, those bacteria growing in the culture will start using up those nutrients. And when they don't have enough nutrients to power their growth starts to slow down. Right? It'll take longer than 20 minutes for cells to undergo binary fission because they don't have enough of the nutrients they need to grow. And then during the next phase of growth, we'll see that the number of cells dying is roughly equal to the number of cells being born. And so the growth will level off, the total number of cells will stay rather stable, and this is what we call stationary phase. When all the nutrients in the media have been exhausted, then we see bacteria in culture go through what's called death or decline phase, where the number of bacteria dying exceeds the number of bacteria undergoing binary fission, and the total number of cells will decrease. So we can watch bacteria growing, not directly because they're too small to see. We can look at those bacteria and watch their growth by using a microscope. And so this is an electron microscope time lapse of some E. coli undergoing binary fission on a dish. So we see one bacteria cell here, undergoes binary fission and becomes 2. Those 2 cells then grow and divide, and after about 20 minutes undergo another division cycle. So we see 4 bacteria here. Those cells grow for a while and then undergo another and another round of binary fission. So you can see the number of cells increases as each one of those cells undergoes binary fission. They form this little lump under the microscope. If we let those bacteria grow and divide long enough, that little lump will become massed of so many cells that we can actually see it. So we can't see individual bacteria cells, they're much, much too small. But if we have millions of those tiny little bacteria cells plopped on top of each other, they form a gooey blob that we call a bacteria colony. Okay. So if we spread some bacteria on a dish and put it in an incubator, the next day what we often see is that we have these tiny little dots, which are masses of bacteria on that plate that arose from one single cell growing and dividing to produce millions of cells that now are so many that we can see them on this petri dish. Right? So a bacterial colony are visible lumps of of bacteria that started as a single cell. Sometimes on dishes we'll see 2 overlapping colonies, so where 2 masses of bacteria kind of grew into each other, right? And so that is an indication that there were actually multiple cells that kind of converge on the dish. You can see on this video that's linked here a time lapse of what happens between this image here and the plate where we get those bacterial colonies on the right. So we said we have these individual bacteria colonies, which are regular round things, and they started as a single bacteria that grew into millions. And then we can also see where those single bacteria colonies kind of converged on each other to form a kind of an irregular mass of bacteria. So if we spread a lot of bacteria out on a plate, what we see is actually what we call a lawn of bacteria. So you can see here on the right a bacterial lawn where we had so many bacteria that all of those cells grew and converged to form this nice little of bacteria on the place. Right. If we grow those bacteria on the plate, we see like a gray mat of bacteria that we call a lawn. If we grow them in a liquid culture, they also spread out to form a nice even layer of growth. We call this turbidity. So one of the things that we can do is we can use that turbidity as an indication of growth. And so as bacteria grow, the more turbid a liquid culture is, the more bacteria there are. But also if we look at growth on the plate, we can see this cloudy portion where the bacteria are growing. We can also see on this particular plate that there are areas where the bacteria aren't growing. And that's because on this plate, we have some antibiotic filter discs. These are tiny little paper discs that have been soaked with antibiotics or chemicals that are poisonous to these bacteria. Right? So you see the bacteria growing here, and we see these areas surrounding the discs where no bacteria are growing. So each one of these discs has been soaked in a different antibiotic. And so I want you, as scientists, to make a hypothesis as to why there is clearing only around some of these antibiotic discs, and why the clearing is different sized on around different discs. So we're going to use this idea of looking for bacterial growth to do in phages what we call a plaque assay. So the plaque assay is going to allow us to detect phages, which are too small to even see with a light microscope in the classroom, by looking at their effects on bacterial growth. So plaques are circular areas where cells don't appear that are specifically due to viral infection. So to do the plaque assay in class, what we're going to do is we're going to mix bacteria and phages. Right? We're going to mix them in a little tube on our desks and incubate them for 10 minutes. And this is going to let the phages attach to and infect the bacteria. We're then going to pour the mixture of bacteria and phages onto a plate with a mixture that we call soft agar. Right? So this is a liquidy agar that will solidify when we pour it on the plate. We're going to pour that mixture of bacteria and phages on the plate and then incubate it for many hours. So if we just put bacteria in that plate, what we would see on the plate is we get a nice even layer of turbid growth because we spread the bacteria all over the plate and they grew together to form a nice even mat. But if we have a single phage infect some of those bacteria, or one of those bacteria on the plate, that phage will produce new phages, lyse the bacteria, and then spread out to the surrounding area. So just like those bacterial colonies form by millions of bacteria piling on top of each other, a single plaque can form by one single phage infecting one bacteria, then producing thousands more phage which spread out in an even area surrounding that initial infection to form a nice round, clear area where all the bacteria are dead. So the plaque assay, if we do this with increasing numbers of phages in the assay, we can see increasing numbers of plaques. Okay? And so if we have a small number of phages, we might just see one or two colony plaques form on this plate. Right? If we have an increasing number of phages, we get an increasing number of plaques, until, just like that bacterial lawn, we actually see those plaques overlap. And this is what we call a web plate, where we have so many phages on that plate that they just eat through all the bacteria. And actually on these plates up here, we can barely see the bacteria because there are so many phages. They just went through the bacteria, and we don't see any plaques because we can't see the bacteria to contrast them against. So the appearance of your plaques is going to tell you something about the number of phages in your sample. But it's also going to tell you a little bit about the phage biology. We learned earlier about the differences between lytic and lysogenic growth of a phage. Now we have some phages that can only reproduce by lytic replication, and those are called lytic phages. And we have some phages that are temperate, where they can reproduce by lysis, lytic reproduction, or by lysogenic reproduction. We have two plates here with two different appearing plaque morphologies on them. So morphology is a word that we use in science to describe how something looks. And so on the left-hand side we see nice round clear plaques, and on the right-hand side we see cloudy fuzzy plaques. Again, I want you to think like scientists and make a hypothesis here. Which of these plates do you think most likely it represents a temperate phage, and which of the plates do you think represents a lytic phage? So this is the tree of life. It's a phylogenetic tree, or a diagram that shows the evolutionary relationships between all organisms. All life on Earth is related to each other, but bacteria are the most distinct group of organisms from the eukaryotes, which is a group that includes us. Eukaryotes are distinguished by the fact that we have a nuclear membrane, or a nucleus, that surrounds our genetic information. Eukaryotes are a diverse group. They include animals like us, but also a number of single-celled organisms, also plants and fungi. Bacteria are prokaryotic organisms. So along with a distinct group of organisms called the archaea, which are also prokaryotes, they lack a nucleus. They're single-celled organisms, and as we talked about before, they fall into two distinct groups. We have gram-positive organisms and gram-negative organisms, so they're distinguished by that structure of their cell wall. In this class, we're going to study bacteria in one particular group. We are going to focus on the actinobacteria. And we're going to use one particular actinobacteria called Microbacterium foliorum, or M. foliorum, as the host for the phages that we isolate in this class. So the reason our choice of host is so important is that viruses have a property called host specificity. They only tend to infect specific host cells. And this is because viruses have evolved strategies to reproduce only in specific cells. So we talked about the lytic and lysogenic reproductive cycles. The viral reproduction depends on the virus hijacking specific cellular pathways inside the cell. And so viruses have adapted to the specific types of regulation and proteins that are inside their specific hosts. For example, over here on the right we have a lambda phage. So lambda is a bacteriophage that infects E. coli. E. coli produce a protein called LAMB, which is inside their phospholipid bilayer membrane. And lambda phage is able to infect E. coli cells because it attaches through its tail fibers to the specific LAMB protein before it injects its DNA. Other bacteria that don't produce this LAMB protein can't be infected by lambda because there isn't that specific interaction between the tail fibers and the protein that let the phage attach and inject its DNA. In fact, if we delete or engineer E. coli to get rid of this LAMB gene, then they become immune from lambda phage infection. Right, so viruses are specifically adapted to their hosts. And this is a property that we call viral tropism. Right. So the phages that you find are going to infect Mycobacterium foliorum because we're going to use M. foliorum as a host in the laboratory. They might be able to infect other actinobacteria, or things that are closely related to, produce a lot of the same proteins and have a lot of the same biochemistry as M. foliorum. but they also might not. It might be that M. foliarum is so specific for the bacteria, or the bacteriophage is so specific for M. foliarum that it doesn't infect other actinobacteria. But regardless, phages don't infect humans because we, in our biochemistry and our cell structure, are way too different from bacteria. So let's talk a little bit about the actinobacteria. So actinobacteria are gram-positive bacteria. Right? They also are rod-shaped. They're bacilli bacteria. So bacilli are a type of bacteria that have this rod shape. So if we look at some microscope images of actinobacteria, like Actinomyces israelii, which is shown here on the right, we have some rod-shaped cells that we can see in an electron micrograph. Or if we use a regular light microscope, we can also see those rod-shaped bacteria cells. Actinobacteria are a diverse group of bacteria, so they live lots of places. They live soil and water, and also inside of animals. So our microbiota, or those microbes that live in and on us at all times, include some actinobacteria. Actinobacteria also have lots of diverse members that do lots of important things. For example, Mycobacterium tuberculosis, or M. tuberculosis, is a bacteria that causes a severe respiratory infection called tuberculosis, or TB. And so it's an important human pathogen. So also we have actinobacteria like Streptomyces. This is a genus with several species that produce a lot of antibiotics. In fact, almost 2/3 of the antibiotics that we use in clinical settings descended somehow from Streptomyces. Right. We also have bacteria that are important for the environment. We have Frankia species. Frankia is a genus of actinobacteria that are important in nitrogen fixing. Right? So we have nitrogen gas in the atmosphere, and nitrogen gas, though, is important. So nitrogen as an element is important for forming lots of the structures inside of organisms. But nitrogen gas isn't chemically available for our cells to use in chemical reactions. These bacteria are able to fix nitrogen and turn it into a chemical form that plants can then use to make their cells, and then we in turn can eat the plants and get that nitrogen into our bodies. Right? So without these nitrogen fixing bacteria, we wouldn't have nitrogen to use for building our cells. So one important thing to note here, when we write bacteria species names, we're going to use a system called binomial nomenclature. Right? We are going to write the genus that a species belongs to as a capital word. Right? So here we have Mycobacterium tuberculosis. And then we're going to write the species as a lowercase word, tuberculosis. Right? So that is going to be capital M, lowercase t, Mycobacterium tuberculosis. They're also going to use italics when we type out these binomial nomenclature names. We also, if we want to, are allowed to shorten the species by abbreviating it. So instead of writing Mycobacterium foliorum, we can write M. foliorum. So capital G, period, lowercase s, species. So Mycobacterium foliorum is a bacteria that was isolated from a sample of grass in Germany. Right? There's nothing super important about M. foliorum in itself. It's gram-positive. It's mostly aerobic, and so it requires oxygen to grow. It lives in the soil. Graham Hatfull, who is the scientist who is one of the creators of the C. phages program, in his lab, he has, for many years, studied the actinobacteria Mycobacterium smegmatis, which is a close relative of Mycobacterium tuberculosis. And he studied M. smeg because this bacteria is a good laboratory organism to study in relation to this pathogenic M. tuberculosis bacteria. So M-SMEG was the first C. phages host. But after looking at all the diversity in M-SMEG phages, C. phages decided to use other hosts to increase the diversity of viruses that we look for. They had some criteria for that, because C. phages is a program for undergraduate students. Those bacteria needed to be safe to work with in a lab. They needed to be able to grow on common laboratory media that we use to grow bacteria. They need to grow relatively quickly so that we don't have to wait weeks and weeks for our bacteria plates to show any difference. And they also need to be susceptible to phage infection. So we've used M. fluororum in our laboratory and have had a lot of success with it. And so we're going to use that bacteria in this course. But other C. phages programs have had a lot of success with other bacteria. Think like a scientist and come up with a hypothesis. Why might classes in different regions of the country, so C. phages is a nationwide program, have success with different bacteria hosts? So lastly, these are some important concepts for studying. When you study for the quiz, just like in lab where we're learning through doing things, we want you to engage in some active learning. Right? So sketch the general structure of a caudiovirales phage, right? And then go through and label the parts. Don't just sit there and look at your notes and try and memorize it. Try and recreate that structure for yourself. Pay attention to the details and then compare your drawing to the things that you see in the slides. Right? You can do the same thing with a lytic cycle of reproduction, so draw a diagram. Right? Write a detailed description of lysogenic reproduction and explain what conditions might be important for lysogeny. Right? You can just do the same thing for bacteria cells, and also do a little bit of math. For example, if a species of bacteria takes about 30 minutes to grow through binary fission, if you start a culture with 10 bacteria, how many do you expect to be in the culture after 2 hours of incubation? So those are some simple things that you can do to study effectively for the quiz.